editing tools or gene editing tools that are being used and being touted as the solution to all of our crop problems, yet again. Of course, old style transgenic GM was supposed to be the solution to everything because that was precise and predictable. But now it's that was clearly. But now they're saying no, that's not precise, that's not predictable. But the new gene editing is. And I hope by the end of this, what I will hope to show you is that that these tools definitely produce GMOs, contrary to what is being argued, and that therefore they need to be regulated as GMOs minimally under the current law, the way old-style transgenic GMOs are. So what are the genome editing? They basically, the key tools divide themselves into two categories. One is what's known as oligonucleotide-directed mutagenesis, and the others are the site-directed nucleases. Under the site-directed nucleases, there are three key tools that have been developed over many years. There are the zinc finger nucleases, the TALEN, and the one that you've all heard about, because that's what's in the news literally every day, which is the CRISPR system, the CRISPR-Cas, or others, that system. On the oligonucleotide-directed mutagenesis, I mention that specifically because there is one commercialized GM crop grown now that has used that, and that is a variety of canola, oilseed rape, that was modified to be resistant to a herbicide called Clearfield. So what this method was used to do was basically tweak one enzyme in the plant so that now it was not blocked by the herbicidal molecule, and so the crop will grow in its presence. And the way this works is, just briefly, you've got your double-stranded DNA molecule. Actually, I'll do it this way. Double-stranded DNA molecule. What you do, you introduce into the plant cells growing in the laboratory a small piece of DNA, what's known as an oligonucleotide. And this small molecule of DNA is homologous. It's exactly the same to a particular target in the DNA of the host plant, except for one or a couple, very few, one or two base unit changes. And through mechanisms that are still not clear, in a few cases, few rare cases, this foreign piece of DNA gets incorporated, exchanges with the native DNA of the plant. So at the end of that, you end up with a plant genome which now carries this alteration that you have that was introduced into it through exchange with this small oligonucleotide DNA molecule. And this is the procedure which the company Cybus used, which their trade name for this procedure is called Rapid Trait Development System. And they use this to generate this variety, and apparently they've got other crops as well, and altered using this method to produce this Clearfield tolerant variety of canola, which is being grown commercially in the USA now, maybe Canada as well. So that's that. But the vast majority of what you're going to hear now is using these site-directed nuclease genome editing tools, and of course especially what's really captured the imagination of everybody. You're going to hear virtually almost exclusively the use of the CRISPR system. And the reason why for that is that it's the smallest, it's the most compact and easy to use, basically. It's in a way no better than the older versions that are out there, the zinc finger nucleases and the TALEN, but it's so much easier to design and to manufacture and to use. Now, obviously, I don't have time to go into the different molecular structures and mechanisms of how they work each individually. That's not really necessary for the concepts we need to take on board today, because at the end of the day, all three of these result in exactly the same outcome. And what I'm going to describe to you now is the classical use of these genome editing tools. They have the CRISPR system in particular. That's now been adapted for several other types of uses in addition to what I'm just going to describe to you. But I'll start with the classical one, which is they're using them, which, as the name implies, site-directed nuclease. What does nuclease mean? It means an enzyme that cuts nucleic acid, in particular here, DNA. So what happens is, basically, through any of these three, to say, admittedly, they are quite different in their structure, but at the end of the day, all of them result in exactly the same outcome, which is the important point you need to take home with you, which is that what they do is, at a predetermined position in the DNA, they bring about what's known as a double-strand break. Basically, double-strand break, DSB. And what that basically means is that they cut the DNA straight across at a predetermined location. It's normally a region. It's not normally, it's not a site necessarily. There's some kind of wobbleness in the area where they work that will bring about a cut within a short region, but they cut straight across the DNA. So you end up now with a DNA molecule in the cell at this predetermined location, which is cut straight across the middle there. Oh, straight across the double helix. Okay? So, what happens after this, after these nucleases have done their job to cut the DNA, everything that happens after that happens completely independently of them. And this is another point to bear in mind, an important mechanistic point of how they work. So what happens after the CRISPR, say, has cut the DNA? The CRISPR doesn't do it. It's out of the equation. So all of this talk that you hear, oh, CRISPR does this, CRISPR does that, that's actually wrong. Technically, it's wrong. All that CRISPR does is to cut the DNA with its associated nuclease. All right? So after the double-strand cut has happened, what happens is that basically alarm bells go ringing inside the cell. Double-strand cut DNA is not a good thing for the health of the cell. So the cell tries to repair it. And there are two mechanisms by which the cell will try to repair this double-strand break. The first one is by far the easiest, simplest conceptually, and for it to actually happen, and which is what takes place most of the time. And that is what is known as non-homologous end joining. What that basically means is there are enzymes in the cell that recognize, oh, I've got broken DNA ends here, I've got to repair them. So there are enzymes that basically are ready to simply rejoin the two broken ends. But this process, again, is not complete. It's not simply a way of, oh, the three ends are just joined back together. Otherwise, you just end up back where you started from, and that's not the idea here. You would end up with something different. So non-homologous end joining is not a perfect procedure. In the process of the DNA ends joining up, you either lose a few bases or you add a few bases. In the process of rejoining, you lose some or you add some. These are, in other words, you result in the formation of what are called indels, or insertion deletions. What that would result, of course, is because you're either losing or adding bases in that region, you're causing a mutation. You're not getting back what you started from. You're actually altering the base sequence. So because of that, if you target the middle of a gene, if you target the middle of, say, a coding part of the gene, the protein coding part of a gene, you will actually disrupt that gene function and you can eliminate it. You can destroy a gene function through this process because you're inserting bases there that disrupt its reading frame, its language. The information content is basically ruined. Okay?
So is that all of the time? I'm not sure.
Through this pathway, you always get indels. Either in our experience, it's deletions, but in some cases, you also get insertion of a few bases as well. Okay, so that results in, say, a gene disruption. Okay, Now, and in some cases that's what you would want to do, and just destroy a gene function. The other half is much more, the other way of repairing that break is more complicated, but useful in when you want to actually change, rather than destroy a gene function, is to alter it. So in this situation, what happens is that you've got your double-strand break DNA here. Then there is another class of enzymes that undertakes what is known as homology-directed repair. So what does that mean? It means that basically those enzymes use a template DNA that is virtually identical in sequence to the damaged area here, and they're able to exchange the DNA, similar to the oligonucleotide-directed mutagenesis here. But you have a double-stranded DNA here that is of a same sequence as where the break is. And what the enzymes do is they will exchange the DNA of this foreign template that you've incorporated with the host DNA. But of course you don't want to have the identical sequence there. You want to change it. So what you're doing here again is you've got changes, a few base changes here in the targeting DNA here, so that when you have the homology, because these regions are homologous of the same, when you have the homology, after the homology-directed repair, the outcome is that you've got the host DNA here again, and that region carrying the changes that were carried in by the template.
Michael, could I just ask you?
Yeah.
The template DNA.
Yes.
This the engineers insert.
Yes, exactly. Yes. You're adding. So in addition, when you're engineering these processes, in addition to putting into the cells, say, your CRISPR system that's going to target and make your cut, you also at the same time introduce your template that's going to result in this homology-directed repair and alteration to the target site. So, and of course, I should add that in addition, rather than introduce small changes into a target gene region, you could introduce a completely new gene into that area. You can make in other words, a large alteration. Yeah, and not just, you can literally insert a complete new gene in a predetermined location as well. And it reminds me, there's one thing okay. So is everyone with me so far?
Michael, could I ask you something?
Yeah.
So what you've been saying is basically the cell's repair processes, the two different types.
Yes.
They produce a variety of different end products, right?
Yeah, they can do, depending on how you set up the initial conditions.
Okay. So, I mean, if you're introducing this, your CRISPR system and your whatever change you want to make into, a population of cells in a dish,
yes.
right?
Yes.
You're going to get this variety of different outcomes. So presumably what you have to do is then clone those cells and find the one that you want. Is that right?
Yes, I was coming on. It's
okay.
Good point, David. But yes. Right. So what David mentioned, very important. We'll come into that any minute now. But basically, this is because of this apparent precision of where you make the alteration in the genome of the host plant. This is why there is the claim now, this is precise, when we can predict all the outcomes from this. Old style transgenics, well no, that's not, we said it was precise, but it wasn't really.
Yes.
But now this is, and so you should just accept this. However, there are many flaws to this argument. Let's see, what's the next say? Right, okay. So to pick up on a point that David made, first of all these procedures are still taking place in the laboratory in what is known as plant tissue culture. You've got dishes of plant cells growing, and you're doing this manipulation on the dish. So yes, at the end of the day, only some of the cells in the dish are going to have the outcome you want, and you've got to select for it one way or the other to find the one that has found what you want. Yes.
Just a comment, because I mean, originally the CRISPR system, at least,
Yes.
is from little lower bacteria
Yes.
and archaea,
Yes.
and it doesn't exist in plants. So they first have to genetic engineer the plant to import that system into it.
No, you don't have to genetically engineer the plant to import it in, because there are well-established what we call transfection protocols for introducing foreign DNA into plant cells on the dish. This is how all the old-style transgenic plants were created. You're using exactly the same transformation procedures or transfection procedures to introduce these products into them at the moment. The system is generic, the way it works. Again, I don't have time to go into why
I
a bacterial system can
afterwards.
adapt, but it's a generic system that will work in any organism once you've introduced it into them.
Can I ask, what you're introducing in, is it from a different organism? Or is it from the same
Yes,
plant?
it's from.
No, these are obviously all foreign tools.
Right.
I mean, they're not. But the argument is that in here, especially depending on the outcome that is expected in most cases, that you're not leaving behind any foreign DNA. You're making an alteration to a host gene region, and then there's no foreign DNA present. Therefore, you can't even consider it transgenic. And that's an argument they're trying to make for.
So what you're introducing in to have that effect,
yes,
is not foreign DNA?
Well, it can be. Yeah, these are.
DNA.
It depends. I mean, in the majority of cases so far, the way these genome editing tools are introduced into cells is as genes that get expressed and have their effect, and then they will be lost subsequently.
Okay.
So something could be a potato on one of your lines,
Yes.
and the other could be a snowdrop.
If I can introduce any gene from anywhere with this technology, but I will have it inserted and I'll select for an insertion at a predetermined site. Whereas in the old style transgenics, I had
It's
no
more random,
control at
wasn't
all.
it? Yeah.
It was completely random where the foreign gene gets inserted. With this, the idea is that no, I'm doing everything at a predetermined location. Whether it's altering a few base units of a gene that's already there, or introducing a completely new gene function at this predetermined site. So, but this whole procedure is still a laboratory procedure. Involves introducing foreign genetic or protein material into the cells and then selecting for the final outcome.
You mean CRISPR itself is a foreign gene?
It can be introduced as a foreign gene. CRISPR is a protein plus an RNA molecule component, a combination of a protein and an RNA.
Are they synthetic or are they natural?
They derive from a natural origin, which is it's a bacterial system which has been adapted so that it will target what you want in the organism that you want.
Because nature does its own selection, its own gene editing. That's why we have thousands of different, millions of different species.
You don't call that editing. You call that just natural variation and selection, which is completely different to what's going on here. I'll go on because there's quite a bit to highlight. So that's the first. The other thing, the arguments are you're only altering one position, but the point about all of these systems, and especially the way the CRISPR system is at the moment, because it's the newest on the block, and it has many more teething problems compared to the other two that have been around for quite some time now, and they've been fine-tuned to work more precisely. But these cuts at the predetermined position, that's not where the only place where the DNA is cut. These tools, especially the CRISPR, have what are known as off-target effects. That is to say, they will cut the DNA at not just where you want, where you're intending it to, but because of imprecision in the way the DNA is recognized and the enzyme will cut, you will get cuts elsewhere in the genome. That is a given, right? That will happen. And many of these other cuts will result, in other words, you'll create mutations not just where you want, alterations not where you want, but the alterations and mutations in other parts of the genome of the target cell. And that's inevitable. It's simply a question of how many rather than if you will or not. Some of these other alterations could result in no change in the phenotype, in the characteristics, but others will. We have to remember that these tools are aiming to disrupt gene functions. There is quite a possibility that they will identify similar gene regions elsewhere and they can disrupt them too. So that's another one obvious level of actually uncontrolled, unpredictable outcome from this. That's one, the fact that you will have these off-target effects. The other place where you can get unexpected outcomes is actually from the altered gene product itself. The most obvious here is that if you change the activity of an enzyme, like here with the seedless canola here, where it was made tolerant to the Clearfield herbicide, if you change an enzyme active site so that it's tolerant to a herbicide or gives it some other characteristic, you will alter the activity of an enzyme. Now enzymes, they are biochemical engines, and if you change the even subtly the activity of an enzyme, it can start carrying out reactions that you don't intend it to. So in addition to getting the outcome you want in terms of enzymatic activity, your enzyme could end up changing biochemistry in other ways as well. So, and the extent of the deletions, this is a new finding. The extent of the deletions here with indels before it was assumed, it was literally assumed, that the length of deletion or the amount of insertion was just a few bases, say up to about 10 bases, 10 base units, 10, 11 base units. But actually, some people who've gone out and actually done the experiments properly, you can have deletions ranging up to several hundred. In other words, when you get the cut in the repair through the non-homologous end joint, you can lose hundreds of base pairs. In other words, you can wipe out the function of just one gene, but multiple genes as a result. The problem with the way where things can sneak through the production of in this case, a GM crop, say, but you have to remember this technology has been used to alter farm animals as well, like getting rid of the horns. Remember, that's hornless cattle. They're coming from this CRISPR technology. What was I about to say? Yeah, you can lose several hundred bases and alter multiple gene functions. And the reason why things were missed in the past was this. When people go to look for off-target effects, they use computer programs, computer algorithms, which search within the genome of the DNA sequence, the base unit sequence of the organism you're trying to alter, and see if they can identify similar stretches of DNA to the region which you're trying to alter. And they say, oh, yeah, the double-strand DNA cut may happen here, where you want, but it may also happen here, 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 and here as well, because of the similarity in the DNA base unit sequence. And so what people do to evaluate off-target effects is that they just go and look at these computer-predicted regions, assuming that the computer program is unfailing. Big mistake. Because now we know when people have actually gone down, looked properly, following a CRISPR genome editing experiment, and what you do is rather than look at these sites only where the computer predicts that you're going to get possible off-target effect, you just sequence the entire genome. Sequence every base, and when you find that is that you will find many off-target effects that the computer program did not predict. And as a result, things can be missed. And a marketed product will have mutations in it that were never found because the evaluation was not conducted properly. Every scientist, the papers that have been published now that have done what we call the genome-wide sequencing following CRISPR editing, all of them have said, we better do whole genome sequencing so we get a complete picture of where the off-target effects are, because their experiments are clearly showing that there are ones that are not predicted, and also their extent. These indels, again, the analysis that was done was simply inadequate to detect these larger deletions that could take out a bigger gene region and lose one or more gene functions rather than altering one gene function at one or more locations. So these are why these tools are not as precise as they were portrayed, especially the CRISPR system, and why they need to be looked on with great caution. So, but what I want you to take, I hope what the impression you're getting from this as well is that when you look at these technologies, any of them, including the ODM here, from a procedural perspective, they're clearly a GM process. There's unquestionably, procedurally, this is not something that's going to happen out in the field in nature or in a breeding experiment. Impossible. They're clearly GM procedures. But what does the industry do, and academia that's advocating this, what do they do to detract attention from that? They say, don't worry about how we generated the final product, look at just the final product itself. Look at what we produced. Oh, you see, we've only changed a few bases in this position. Look at that final outcome rather than the procedure. And then they will argue that these small base unit changes that you've made, well, there's something that could happen through random mutation out in the field. So why should you worry? Well, first of all, that's our hypothesis. There is absolutely no evidence at all that a small base unit alteration in a crop through CRISPR has been found pre-existing out in the field. If it has been the case, go out and find it and show it to me. Survey 1 million canola plants or whatever you want, or maize plants, and show me that what you generated in the lab actually exists out in nature. That has not been done. These are purely fantastical arguments, in my view, that they're using to try to detract attention from the fact that procedurally they're clearly GM, and the end products are clearly GMOs and should be regulated as such. I'll leave it at that. I think, because I've run out of time. But as I say, there are adaptations of this technology now that are able to change DNA not through this double-strand break, or alter gene function, DNA sequence and gene function, without relying on a double-strand break. But those are other developments, and I don't have time to actually go into them. But all of these will be prone to the off-target effects and at predicted and unpredicted sites, and therefore with unknown consequences in terms of the composition of the plant and its consequences for environment and health. So that's that. How's that?
It's fascinating.